of this webinar. So I'd like to turn it over for some questions if we have time for some.
All right, ladies and gentlemen, we have now come to the question and answer session. I shall read questions from the audience for all to hear before Heather responds. If we are unable to respond to your question during this time, please feel free to reach out to us by email, and we will do our best to respond to each in a timely manner. Okay, Heather, our first question is, as you talked about panel testing in both of the clinical case examples you discussed, do you feel that exome testing is likely to eventually replace panel testing?
That's a great question. I think we get this question a lot in the field as well. And, you know, right now, I think that panel testing and exome sequencing each have their own special place in the clinic. There's certainly some genetic conditions where a panel might be the most useful approach, and certainly other areas where exome sequencing is more useful. There's obviously some difficulties with panels, as we talked about. One of them is that the genes change quite often, and so there's difficulty in keeping up with the changing genes on panels, even in one laboratory or in other labs. There's different genes on different panels across medical laboratories, and I think that's something that might be really difficult to keep up with in the actual clinic. I think the other side of it is that exomes, you know, of course, have their own inconsistencies. There's certainly, as we talked about, coverage gaps in exomes, but also coverage gaps in next-gen sequencing-based panels as well. And so, you know, we really focus on trying to sort of bridge that gap with our augmented exome. And I think in those cases, if we're able to supplement these areas where there's poor coverage, then that might be a good argument for taking the place of panels in the future, but I still don't think we're quite ready for that right now with some of the panels out there.
Thank you, Heather. The next question we have is, what is Personalis' policy on reporting secondary findings on clinical samples?
Sure. You know, just like every other laboratory, we do agree to report secondary findings according to ACMG's recommendations. We do take a somewhat different approach in that we have it as an opt-in part of our standard consent form. We do testing on the proband only, so ACMG findings are not reported for family members in the case of a trio analysis. So those results are reported on the proband only.
Thank you, Heather. So the next question I have for you is, how is Personalis able to increase coverage of difficult to sequence regions? What do you do differently to achieve this?
So another great question, and one that comes up commonly in the field as well. You know, what we do is we actually have a proprietary method that includes proprietary sample prep, enrichment, and sequencing to supplement targeting in those difficult to sequence or those regions where there's poor coverage in standard exomes. We actually combine that enhanced capture with DNA that's captured from a standard enrichment protocol, and we do that prior to sequencing. So while I can't say exactly what it is that we're doing, a lot of it is really improvements prior to the sequencing end.
Thank you, Heather. So we have time for just one last question for today. So it is, why is there so much variation in coverage of the medically interpretable genome across the different exome platforms that were included in this paper?
Sure. You know, so one of the things that we pointed out earlier on in the presentation is that, you know, we tried our best to normalize samples to have the same average coverage across the complete target region. But even though we did that, you know, what resulted is that the average coverage in that medically interpretable genome, those 5,400 genes, that still ranged quite a bit, anywhere from approximately 95x coverage to approximately 210x coverage in that medically interpretable genome. But as we've also talked about, average coverage really doesn't correlate well with gene finishing. There's certainly some areas that are going to be poorly covered, and some areas that are going to be adequately covered are also covered up above and beyond what you may need in a clinical setting. And so that kind of helps to illustrate some of the differences that you see in the actual design of each of these platforms that we've talked about. Things like probe design and library preparation, those are things that also affect how well each particular region is covered. And, you know, as was evidenced by what we found in our publication, you know, this is really apparent when you start looking at things at a clinical level and testing performed by these clinical sequencing labs, is that all of these platforms aren't designed with a clinical mindset. Many of them are more designed for research or gene discovery, but there are a couple of XM platforms that sort of come off on the top, including ACE, that are more designed for clinical use. And so that's why you see that variation in coverage.
Thank you. We have now come to the end of our time today. Thank you all very much for joining us. We hope that you all found this presentation beneficial, and again, extend our thanks to Heather for leading this informative and interesting session.
Thanks again, Felicia.